So our first speaker will actually represent one of the partnerships, which is the CFAR, Duke CFAR MEDx partnership. And this is Dr. Collier. There's Dr. Collier. So Dr. Collier is an associate professor at Duke in biomedical engineering. So welcome.
Good morning, everyone. Pleasure to be here. Yeah, I'm very pleased to be here to describe our pilot project to you. Again, this was a joint CFAR MEDx, let's see, you seeing the pointer up there? Pilot grant. And this was a collaboration that was, so my lab, I do bioengineering work, biomaterials work. This is a new foray into HIV for our lab. And it was in collaboration with the labs of Genevieve Fouda and Sally Permar. And it was spearheaded by two of our graduate students. So Chelsea Fries is a graduate student in my group. I'm not sure if Chelsea's here. And Juilin Chen, who'll have a poster. There's Juilin over there, who'll have a poster later today. If you want to get into any of the nitty-gritty details, I'm sure he will be happy to spell them out for you. So we work on designing novel HIV vaccines. And I don't think I need to tell many people in this group that that is a important part of HIV research. You know, a number of people are still, many people are still being infected. Even so, over 40% of people aren't on IRT, and even those that are still can transmit the virus from mother to child. And so we, of course, have been trying to elicit broadly neutralizing antibodies, which are the one of the main standards in vaccine design. And no vaccine presently can induce these. As I said, so my group is involved in biomaterials and nanotechnology and protein design and designing particles, gels, fibers for various applications, including tissue engineering. And we've gotten into immunological applications in the past few years. Again, we hadn't worked on HIV. Some of the materials that we build are very highly multivalent, so they have many copies of certain features. They can be antigens, immune-stimulating compounds, specific epitopes, and we thought this would be an interesting application to direct them towards HIV. With respect to multivalency, it's a very important way of driving B-cell activation and other more complex immunological interactions, but one way is by cross-linking the B-cell receptor. And so existing platforms that have been explored so far primarily are based on spherical proteins. There are a number of proteins that have been utilized and explored. Most of them are small but have multiple attachment points for antigens. Here we have one showing eight. There's one projecting out of the screen and one behind copies onto ferritin. And these have been shown to take advantage of this multivalency effect, but it's a fairly limited real estate that you have on a protein particle like this. And so there's a limited number of antigens that can be attached. If you would like to attach multiple antigens, this becomes challenging. When it comes to driving immune response, it isn't just the antigen. There are other factors that are important in incorporating into the material. And you quickly run out of room when you're dealing with small soluble protein carriers. And so our project was about designing a very multivalent vaccine using a platform known as self-assembling peptides and to initially assess the immunogenicity in mice. Asking a few questions about whether or not we can improve antibody titers, whether or not adjuvants can augment any effect that we see, and whether or not we can adjust T-cell epitope content and optimize or improve the response from there. So the platform that we're using is a short peptide. We've in our lab developed a number of peptides. This is one that will assemble reliably. So if you take this peptide, dissolve it in water, and then intermix it with neutrally buffered saline or body fluid, any sort of neutrally pH neutral, high ionic strength, salty solution, it will assemble into nanofibers, right? And so the scale bars down here, they're really quite narrow. This is only around 20 nanometers wide, and they can be hundreds of nanometers long. And the nice thing about this particular peptide is that if you append it with other epitopes of choice, they will co-assemble into integrated nanofibers that also have this similar structure. So here's one that happens to bear the OT2 antigen from ovalbumin, very common immune epitope. And you still get these nice long regular nanofibers. These materials are also self-adjuvanting, so they can evoke quite strong antibody responses even without the addition of additional adjuvant. And so the concept for our pilot grant was, let's try to devise ways to get HIV, fully folded HIV antigens onto these nanofibers. And we've worked through a number of different chemistries. I'll describe the first one that the pilot grant was built on. We've since sort of ratcheted up the project into a more complex, RL1 funded, different antigen conjugation scheme, which I'll get to in a minute. But to start with, we simply took GP120, HIV GP120, and we used heterobifunctional crosslinkers to chemically conjugate the antigen onto the formed nanofibers via a cysteine Q11 peptide that we co-assembled into the materials. Chelsea and Ray, Jui Lin, did a number of experiments to make sure that the chemical conjugation was not altering the antigenicity of the protein, and devised a way to do it so that we retained binding of all these antibodies with similar K-off rates by bilayer interferometry. And then we started vaccinating mice, and we found that without adjuvant, we could raise reasonable titers. This is a prime boost regimen. And interestingly enough, GP120 by itself was fairly immunogenic. Oligomerizing it onto the Q11 scaffold gave it a significant boost in immunogenicity. One of the things we observed early on was that as well as increasing titers, we had better cross-reactivity of those titers against different, so this is the immunizing, this is a little bit of a difficult to sort through data slide. But so we immunize with either the protein or the oligomerized nanofibers with the protein on it. And then we look at antibody responses against the immunizing form of GP120 and other forms of it shown in these columns, right? So if you have good cross-reactivity, you end up with a lot of red being the most, area under the curve antibody titers. So comparing the oligomerized with the unoligomerized, we get a much broader response. And this, we think, is going back to the cross-linking of the B-cell receptor, better germinal center formation, better affinity maturation from B cells. So the next thing we did is we tested this with an adjuvant, and this is using an adjuvant developed in Tony Moody's lab, known as straight SC. This is a TLR7, 8, and mixed TLR9 adjuvant. And this is fewer doses, so this is just a prime boost, and we get a much greater enhanced effect by oligomerizing the antigen on the nanofibers. We also have a better increase in the breadth, right? So we have fairly non-broad response against the antigen by itself. When we oligomerize it on the nanofibers and use straight 8SC, we get a much broader response. So one of the benefits of building vaccines this way, as I alluded to in the setup of the work, is that there's a lot of room on these nanofibers, and you can put other factors in with it as well. The room allows us to alter the density, right? So a B-cell receptor has a specific geometry, of course, and the oligomerization of it depends on what you could envision. It'll be maximally stimulated with a specific organization of that antigen. And so with this system, we can devise high and low loading densities of the material, or we can mess up the oligomerizing domain, so we just have soluble protein with the linker. And in that case, we have, with just a prime boost, a much greater enhancement of antibody titer for a given dose. So this is the same amount of GP120, just clustered together on fewer nanofibers or spread out over more nanofibers. So this gives a engineering access to being able to sort of finely tune this going forward. And that's not something that is really able to be done on a spherical nanoparticle very well, or a spherical carrier protein. As I mentioned, this also affords the opportunity to integrate other factors. So we made a like T-cell epitopes. So we made a Q11 peptide with a flexible linker, and so this is a universal T-cell epitope that will bind most MHCs. We co-assembled this, do the reaction to bind the antigen onto it. So we have an integrated nanofiber with the T-cell epitope and the GP120. Interestingly enough, although in some of our other work with different diseases and different therapeutic vaccines, the incorporation of the T-cell epitope has a huge influence, here it has a relatively minor influence. And we think this is likely because the GP120 itself contains T-cell epitopes that are likely even better and stronger than the Padre. So that was the main focus of our pilot grant. Right now, as I mentioned, we submitted and received a new R01 to take this chemistry and this concept and apply it to different, better, we think, antigens. So here's a SOCIP trimer, and this is work in collaboration with Kevin Saunders and others in DHVI. And so the SOCIP is expressed with a sortase tail. Sortase is an enzyme that will catalyze the conjugation between a short peptide sequence on the protein and a polyglycine linker on this peptide. So we make a SOCIP that is capable of inserting into the nanofibers. This is using a technology that we've coined called beta tail technology that we published a few years ago that allows expressed proteins to insert into these molecular assemblies. So the assembly would look like this with SOCIP trimers projecting from it. And we continue to explore these. We're starting our first rabbit study very soon, and we're gearing up to start our first non-human primate study early next year. So for some scientific conclusions, we've found that the nanofiber presentation does increase antibody magnitude and breadth when we use GP120. This is particularly effective when we use adjuvants, especially this straight SC adjuvant developed in Tony Moody's group. We had a little bit of help from incorporating extra T cells, and we started to really tease apart that the antigen density is a really important determinant. We just recently, in April, received R01 to continue this work, as I mentioned, with the SOCIP trimers and in rabbits and non-human primates. And so if you sum up all the inputs, so this was a joint MEDx CFAR grant that came into our labs. We have this new funding. We just submitted an invited review in Nature Nanotechnology on vaccine design that all of us are authors on. That was a really fun manuscript to write together. It was certainly new territory for my group. Dwi Lin is working on a manuscript that we will be sending in soon. We have IP, so we filed a patent last year. The provisional went in, and we're sending in the full patent next week. It's due next week. We're eagerly working on that. And like I say, we are moving to rabbits and NHP as the work continues. None of this would have been possible without the initial CFAR grant that we received, so I want to express our sincere gratitude for catalyzing not only what I showed on the previous slide, but this group that now meets every other week and has discussions on new work that we never would have considered without this gentle nudge to assemble our group. It's been a delight working with Jenny Fouda's lab and Sally Permar's lab and their members, and a number of collaborators, some of whom I've mentioned already, have been giving us advice and help along the way, including Tony Moody and Kevin Saunders. Bart has been a great sounding board and source of advice for this work. Herman and has nucleated other collaborations with others in this group, including Herman and Nick. So thank you very much. I think we have a minute for questions. Happy to answer any you have.
Joel, that was tremendous. I did notice on one of your early experiments in mice that mouse 2 was a dud all the way across as far as GP120. Did you ever go back and think about giving them then the oligomer, see if they had any reactivity against the oligomer? Or is that...
Against the carrier? Against...
In other words, it got all GP120 and didn't have any actual broadening of reactivity.
I see.
And I was just curious what was wrong with that mouse, and would that mouse have responded to the oligomer rather than 120?
Perhaps. Yeah.
Just curious.
Yeah, we didn't do that. That would have been a good thing to occur. Is it still around? It'll be very old if it is still around. I don't think it's still around. Yeah.
So Joel, you mentioned that your nanofiber formation allows better BCR cross-linking. So I wondered, but you also commented that it has some inherent adjuvant activity when you use the nanofiber form.
Right.
Do you know if that adjuvant activity is simply due to better BCR
I don't think it's
cross-linking
only that.
or
So,
something else?
Yeah, like most adjuvants, I think many things are going on simultaneously. Some of the TLR agonists that are extremely refined, have a precise point that they're interacting with. But compared to something like alum, right, which has a very multifactorial mechanism of action. And I think that's in the realm of what we're looking at here. So the particulate nature of it allows it to be taken up by antigen-presenting cells very quickly and avidly. So you have, from the T cell side of things, you have, promotion of that. We've looked at whether or not, so when an antigen-presenting cell does pick up a particulate, usually something like the inflammasome is going to be activated. We don't see that. We've looked in inflammasome knockout mice. We don't, it works fine. So there are a lot of parallel things going on. We've done in vitro B cell activation experiments, and especially, so we have a complement that we're dosing into this for other projects that greatly enhances the B cell receptor cross-linking and activation. So there are a lot of things going on at the same time. Particulate nature, B cell receptor cross-linking, just high loading of the antigen, right? I mean, any B cell is going to see a large copy number at the same time. Any antigen-presenting cell is going to display on their MHC a lot of that T cell epitope that's co-assembled. That's what we have right now. It's not a simple, straightforward single factor thing right now. I think we have one more, if there's time. Is there time?
Thank you. Did you see any neutralizing antibodies using the nanofiber and the GP120?
We haven't looked at neutralizing antibodies yet.
It would be interesting to see if how those neutralizing antibodies, if you get any, compared to the SOSIP tetramerized with the nanoparticle.
I agree, right. Thank you.